Okay, so we will get started for today's research seminar. Our speaker is Dr. Charles Cohan. He serves as the data science manager at the Gateway Institute for Brain Research. His work is centered on leveraging advanced computational techniques in personalized medicine to address complex neurological challenges. Dr. Cohan earned his Bachelor of Science degree in neuroscience from the University of Michigan in 2009. He then pursued his PhD in neuroscience at the University of Miami, completing it in 2015. During his doctoral studies, he investigated neuroprotective mechanisms to prevent injury following cardiac arrest and stroke under the guidance of Dr. Perez Pinzon. Following his PhD, Dr. Cohan worked as a postdoctoral fellow at the University of Miami for 3 years, focusing on models of white matter injury. In 2018, Dr. Cohan joined the Gateway Institute for Brain Research, where he initially conducted both bench science and computational work. Over the past 7 years, he has progressed to manage the data science department and leads several research initiatives. His current research initiatives include integrating computational modeling, bioinformatics, and in vitro testing to develop innovative treatments for Parkinson's disease, aligning with the Institute's mission to find a cure. Today, he'll be sharing a presentation titled Personalized Medicine Approaches in Parkinson's Disease. Without further ado, Dr. Cohan, everyone.
Thank you very much. All right, so today I'm going to go over a background, just discussing Parkinson's disease, giving specifics about the disease and talking about current challenges in Parkinson's disease research. I'm going to talk a little bit about the goals of the Gateway Institute for Brain Research. I'm going to go into details about our personalized medicine-based approach to tackle these challenges involving Parkinson's disease, and then talk about future directions of where we want to go. So to start, Parkinson's disease is a debilitating disease that impacts millions of people worldwide. This is from a recent study looking at the prevalence and the predicted prevalence of Parkinson's disease up to 2050. And currently where we are right now, the prevalence of PD worldwide is over 12 million people and is projected to jump up to 25 million people by the year 2050. So this is something that's not going to go away. It's going to increase. If you break it down by countries that are the most affected, right now, the United States is the third most prevalent country, and also India and China have very high prevalence of this disease, and particularly in China, the amount of people with Parkinson's disease is going to explode as the population, the average age of the population increases. So again, it's not just a problem here in the US, it's a worldwide problem that's only going to continue to grow. So what is Parkinson's disease? It's symptomatically, it's most characterized for motor deficits. So people have tremor, postural instability, bradykinesia, slowness to initiate movement, or rigidity, or dysarthria, trouble speaking. However, it's not just motor symptom or motor dysfunction or motor symptoms. There's also loss of smell, anxiety, dementia, and a number of other non-motor symptoms that arise as well. And one thing that's really important to point out here is that there's no cure for Parkinson's disease. It's something that just continues to progress throughout the life of a patient who develops it. When you ask patients what it is that they want to treat about Parkinson's disease, what I find interesting, this is a recent publication where they did a survey of what is the priorities that patients want clinicians and scientists to tackle in terms of treating Parkinson's disease. There's no one answer. So this is a list of symptoms here, and with what they want, there's a primary priority, a secondary or tertiary priority. And what you can see here is that nothing goes above 20%. So there's a wide array of symptoms that people want treated, and there's a group of motor symptoms that people want, or tremor being one of the highest, but again, lots of diversity here in different motor symptoms. However, there's again these non-motor symptoms that are also very important to people to treat, including fatigue and energy, sleep problems, and cognitive function. So this isn't just when I think traditionally when a lot of people think of Parkinson's disease, they think of the motor dysfunction, but it's not just that. There's a lot more that's going on. So what are people doing to develop treatments for Parkinson's disease currently? This is a recent review looking at the clinical trial pipeline as of 2024, and it's looking at phase 1, phase 2, and phase 3 clinical trials. And there's, if you look at this pie chart here, what you can notice is that this bottom half of it here, this is just symptomatic therapies. So a majority of the therapies that people are investigating for Parkinson's disease are addressing symptoms, which is important for the quality of life improvements that were mentioned on the previous slide, but it's not going to fix every problem. So it's nice to see that there is a good amount of disease modifying therapies that are actually looking to cure the disease. However, there's less, and if you look at the number of trials that are actually in phase 3, there's only 3 here, with one of them, I think, very recently having somewhat negative results come out in a clinical trial. There's a lot of diversity in the type of treatments that are here. Some interesting ones that you might notice here, these are GLP-1 agonists, so might have heard of Ozempic, or the diabetes or weight loss drug is one potential treatment here. Another one is alpha-synuclein targeting drugs. So there's a protein that's very important in the disease pathology of Parkinson's disease called alpha-synuclein, and there's a whole category of drugs here. But that's not the only thing here. Again, there's a pretty diverse array of therapies, and I think that speaks to the underlying nature of the disease being somewhat diverse, which I'll get into. So what goes wrong in Parkinson's disease on a cellular level? This is a complicated question, and there's no one answer here. So as I mentioned on the previous slide, there's this protein called alpha-synuclein, which misfolds and forms these fibrils and oligomers, so these like clumps of misfolded proteins that ultimately collect together and form these structures called Lewy bodies that are within cells. And these Lewy bodies help contribute to the disease. They can help trigger lysosomal dysfunction. So there are systems that are designed to help destroy proteins that are faulty. You can experience problems with those mechanisms, which can lead to further buildup of the Lewy bodies. So there's this interplay between these two things. And there's known genetic risk factors in both lysosomal metabolism and also the proteasomal ubiquitination system, a number of genes involved there. So there's this interplay between these symptoms that seems to worsen the amount of alpha-synuclein fibrils and oligomers and Lewy bodies that you get forming there. In addition to protein dysregulation, there's also problems in mitochondrial dysfunction. So there's a number of other known genetic risk factors associated with mitochondrial function, where the structure of the mitochondria is damaged or not repaired appropriately. And this is also another potential cause of Parkinson's. Furthermore, oxidative stress is also a big factor in these cells. So the types of neurons that are involved in Parkinson's disease are called dopaminergic neurons. And dopaminergic neurons are highly bioenergetically leveraged. So they sort of live on the edge. They require a lot of energy due to certain properties of dopamine, where they need to produce a lot of energy, and they also produce a lot of reactive oxygen species, which makes them very susceptible to oxidative stress and damage. So again, this is another contributing factor to the sensitivity of these cells. Also, inflammation is thought to play a role, and amyloid plaques, which is another hallmark of another neurodegenerative disease, Alzheimer's, can play a role in increasing inflammation and activate microglia and other inflammatory mediators. And finally here, I just want to highlight this other protein here, tau, which is another hallmark of the disease, where you get these neurofibrillary tangles of hyperphosphorylated tau, which contributes to the inflammation, the oxidative stress, and the mitochondrial damage. So what you can see here is that it's not simple. There's a lot of different pieces here of the cell that are dysfunctional, that cause problems in the case of Parkinson's disease. So what actually goes wrong in the brain? Well, within your midbrain, you have a circuit called the basal ganglia. And the basal ganglia has two sort of subcircuits within it. One is referred to as the direct pathway, and the other one is referred to as the indirect pathway. And these two pathways receive input from a region of your brain called the substantia nigra pars compacta, which is depicted here with this purple arrow going away from it. In the case of Parkinson's disease, what happens is you see damage to the substantia nigra pars compacta, and this actually removes inhibition from both the direct and indirect pathway and causes dysregulation here. And this basal ganglia circuit is very important for motor function, particularly initiating motor function. So when you can't regulate these two pathways, you see this dysfunction and you get trouble with motor symptoms. As we talked about before, it's not just motor symptoms that you see with Parkinson's disease. So how is that the case? Well, alpha-synuclein, this protein that you see these aggregates of forming in the substantia nigra, doesn't just stay restricted to the basal ganglia. It actually spreads throughout your brain and can, expand out to the cortex and other areas as well. And there's actually potential differences in where this is coming from. We're not exactly sure which of these hypotheses is right, or whether the alpha-synuclein aggregates are actually happening in the brain first, or if they go to you're eating something, or you have bacteria within your gut that produces alpha-synuclein, and then it migrates up to your brain, up to the substantia nigra, and then out to the cortex. So it's possible that in some combination of people, one happens, and in another one, another happens, and people think that there might be differences in the symptoms you experience depending upon where it originates from. But what's important to note is that we're not exactly sure where this dysregulation and dysfunction of alpha-synuclein is coming from. And one final point I just want to highlight about Parkinson's disease here is that there are a known number of genetic isoforms of Parkinson's disease and genes that are linked to Parkinson's disease. So there's over 18 PARC genes, and if you have these genes, you're much more likely to develop Parkinson's disease. And one thing, when you look at these different genes, the types of mechanisms they're involved in are very diverse. So, for example, PARC1 is alpha-synuclein, this protein, which is actually a synaptic protein in nature. You have ubiquitin cyclohydrolase A, which is related to the proteasomal ubiquitin system. So again, degradation of proteins. You have DJ1, which is important for mitochondrial fission and fusion. And you have VPS35, which is a cellular trafficking protein. So just highlighting again, there's lots of potential genetic risk factors, and they're spread across many different mechanisms. And in fact, over the last 25 years, after there's been a number of these large GWAS studies that have come out identifying genes that are risk factors for Parkinson's disease, and they're up to identifying, I think, over 100 different genes that might play a role in risk for Parkinson's disease. And even with these 100 genes, they say it only accounts for 11 to 15% of the heritable risk of Parkinson's disease. So most Parkinson's disease, despite all these genes being linked to it, is sporadic, where we don't know what the underlying cause is of what's triggering it. So if you want to look at all of the networks that have been implicated, this is actually not all of them. It's just a subset of networks that are thought to be involved in Parkinson's disease. You see a pretty complex picture emerging of these different networks, and there's many pieces here and many potential things that could go wrong. All right, so just to summarize the review, there's no current cure. It's a debilitating disease that affects many people, and the prevalence is only going to increase over time. We don't really know the underlying cause. Many think tau and alpha-synuclein are involved in the pathology. We don't really know where it's coming from, external or internal. We know there's many different genes involved and many different pathways involved. And so this is a challenging problem. How do we solve it? And so now I'm going to talk a little bit about the goals of Gateway. So this is taken from our website just to sort of describe who we are. So the Gateway Institute of Brain Research is an international research institute that combines computational modeling, laboratory, and clinical research. Our goal is to identify therapeutic strategies and develop advanced treatments for Parkinson's disease and other neurological disorders. Our mission is to cultivate collaboration and innovation to develop treatments for people living with neurological disorders, and we want to revolutionize patient care by integrating scientific approaches and translating medical research discoveries into cures for neurological disorders. All right, so what does that mean for this complex disease? So let's turn back to this web of networks, this complex web of networks that we have here. And when we look at this, what we see is that you might have a problem, say, in like proteasomal breakdown. You might have a problem in mitochondrial fission or fusion, or you might have a problem in alpha-synuclein levels or regulation in some manner. And so, when we look at this, it's pretty clear to us that Parkinson's disease isn't just one thing. You know, you can have lots of different things go wrong in these mechanisms, and the end pathology might look similar, but with the underlying cause, it's probably not going to be just one thing. And because of that, you know, we ask this question: will any one treatment actually cure everyone who has Parkinson's disease? To us, you know, it doesn't seem the most likely scenario. So, what's our philosophy? It's that Parkinson's disease isn't just one problem. And the best chance we have to solve Parkinson's disease for an individual patient is to look at what's wrong in that individual patient. And so if you think about recent cancer biology research, you know, it's progressed to this point where there's this emphasis on precision-based medicine in cancer biology, where you can take a tumor from a patient, you can do genetic profiling on the tumor, and you can see if there are certain hallmarks there for certain treatments that will be more effective for treating that tumor. And to us, you know, this sort of focused approach on looking at what's wrong in the patient and devising treatments around that seems like your best chance to try to fix what's going wrong with an individual. So people have theoretically been thinking about this in the concept of Parkinson's disease, and it would look something like this, where you have different molecular pathways that are, you know, dysfunctional. You have your heterogeneous Parkinson's disease population. You separate them out into etiological subtypes, and then you manage those subtypes. So you devise a treatment that's going to fix immune system for people with an immune system problem, right? So seems relatively straightforward. The hard part is how do you figure out these different subtypes? How do you devise the treatment for it? And so that's where we get into our personalized medicine-based approach. So it's 3 steps, really. The first thing we want to do is we want to get data from patients that reproduces what's wrong in those patients as best as possible. We want to use computational approaches to come up with fixes to what's wrong. And we want to test those therapies to see if they're effective in fixing the networks and dysfunction that we see. So first, figuring out if we can, what should we use from patients in order to determine what's wrong. So the easiest thing to do would be to get, you know, blood or skin from a patient in order to determine if we can utilize that to really get a good profile of what's wrong. So some recent research has looked at both of these tissues in patients with Parkinson's disease. And what they found is that blood and skin actually show very different profiles of dysregulation within them compared to one another. And when you actually take those 2 tissues and you compare them to the substantia nigra of patients, it actually is very different than what you see in the substantia nigra, and there's virtually no overlap between dysregulation. So to us, this is sort of a red flag that if we use these 2 tissues, perhaps we're not really going to capture exactly what's wrong with these patients. So what can we do to actually get a better view of what's wrong with these patients? Well, more recently in the research, there's been 67 studies where people have developed essentially inducible dopaminergic neurons for Parkinson's disease patients. And so what they do in this context is they start with either skin cells or blood cells, and they convert those into inducible pluripotent stem cells, and then convert those into dopaminergic neurons. And so this is the benefit of working with neurons in the end. This has been done many times for a lot of the different genetic isoforms of Parkinson's disease. And when you look here at the bottom, this is the sporadic group, the group that doesn't have genetic risk factors associated with it. So it has been done to a lesser degree also with these sporadic PD patients as well. And so why this is particularly attractive to us is when you look at the actual neuronal phenotypes of like cellular dysfunction that you see here, these cells actually reproduce the injury that you see in the Parkinson's disease patients. So for example, they can show alterations in regulation of tau, or altered mitochondrial respiration, or neurite growth, or oxidative stress response. So there's something about when you make these into neurons, they recapitulate the disease in a dish to some capacity. So to us, we feel like this is the closest we can get right now to brain of these patients, because we can't take brain cells from these patients because that's a little invasive. So this is the best we think we can get. All right, so our lab can produce inducible dopaminergic neurons from patient blood, and a lot of this work that I'm going to refer to from here on is from the in vitro group in our company, which is headed by Madeline Cox. So what we do is we take blood samples, we isolate PBMCs from those blood samples, we convert those into inducible pluripotent stem cells, and then into inducible dopaminergic neurons. We've taken these dopaminergic neurons and we've run RNA-seq, and then it's some additional confirmatory steps to actually make sure that we can produce these dopaminergic neurons. And in the RNA-seq results, what we've seen is that we've examined a number of dopaminergic markers, including tyrosine hydroxylase, which is this critical step in the production of dopamine. It converts L-DOPA into dopamine and is necessary for dopaminergic neurons to actually produce dopamine. And we see this massive upregulation in these cells after 60 days in culture for these neurons. And additionally, it's not just tyrosine hydroxylase, there's other proteins from the same pathway for production of dopamine, and then also an upregulation in alpha-synuclein, this key protein that's involved in PD. So we have good evidence that we can actually make these dopaminergic neurons. This is a immunofluorescence image of tyrosine hydroxylase, and also DAPI, so just a nuclear stain, as well as looking at tyrosine hydroxylase. And what you can see here is that there's these sort of like neurite projection highways that you can see within the culture where there's lots of TH expression there. So it aligns with the RNA-seq results. And they also did ELISA work. This is again provided by Madeline and her team, where they showed that these cells are actually producing dopamine as well. So they have the markers for dopamine, they have TH present, and they're producing dopamine. And additionally, one interesting finding is that when they compare a non-PD cell line to a PD cell line, there actually was elevated ROS found in the PD cell line. This is an N of 2, so it's not statistically significant, but it's trending towards there and currently being worked on. So it looks like we're seeing the disease recapitulating this oxidative stress in the PD cell line. Additionally, the lab is working on extending this process out, not to just use the IDANs, but also to use organoids. So this again is 3 markers that are important. So this is alpha-synuclein again, and you can see there's alpha-synuclein present within these organoids. There's beta-3 tubulin present, which is like a neurite outgrowth marker. So there, we have neurons with extensions, and again, tyrosine hydroxylase is highly expressed within these organoids. And again, preliminary evidence is showing that at day 41, these organoids are producing dopamine again. All right, so where does this leave us? So we think now, we're able to get cells, we'll start from blood from patients, convert it into these dopaminergic cells or organoids, and produce injury that we would see in the patient. So the next question is, how do we come up with an approach to fix what's wrong in these cells? And so this is where we turn to a framework known as logic modeling. And so a lot of this, our work originates from work done by Nancy Klimas and Travis Craddock, who's the lead author on this publication. And so the idea is that you can take this is looking at the HPA axis and its involvement in Gulf War illness. And so what they do in this paper is they take this HPA axis and they convert it into what's called a directed graph. And a directed graph is something that consists of nodes, so these circles, and then these edges, which are either marked green for a positive regulatory effect or red for a negative regulatory effect. And so the idea is, if you know all the pieces of a network and you know how all these connections play out, and you know what the starting state is for that patient, you can sort of simulate and predict what's going to happen to that patient over time. And one added thing you could do is you could then simulate what happens if you change one thing. So for example, let's say you know that the patient has high cortisol and you want to see what the effect would be of reducing cortisol in the patient. Well, you could simulate lowering cortisol and then letting the simulation play out and seeing what happens over time. So within this publication, they did this, they performed all these simulations, and they were able to come up with these treatments for Gulf War illness, where you target specific nodes within this, and then you wait and you target another node, and they find out what order of this therapy works out the best in order as to treat this disease. One important thing to note here is that they did this originally for this HPA axis model. It's not just limited to that. You can take any network and convert it, as long as you know the nodes and the edges here, you can convert it into a directed graph for anything. So this is a later publication by the same group looking at neuroinflammation. And so here in this network, there's microglial, you have microglia, the effects of microglia and pro-inflammatory cytokines and astrocytes and various growth factors on neuronal activity and the interplay between all these things. And I just to sort of like drive home this concept of like how this really works, I just want to talk a little bit about like the underlying concept here. And so the idea here is that if you look at, say you have something that looks like this, right? And what this essentially is it's a graphic, it's a depiction of health within a patient, right? Of all possible health of states of how a patient could be doing with a network. And so what I mean by that is like, if you look at this ball, right, this ball represents the current state of the patient. And then if we had with that network we just looked at here, we could say that this is a completely healthy state here where everything is normal. So white here on this picture on the side here indicates that it's normal. So this sort of like basin, you could call it this little basin in the very bottom of it where this ball is a normal basin. For any individual, what can happen where you have like little perturbations, you get a cold, you get sick in some way, you have these little perturbations that can kick you around this basin. So you might have little changes in some of these nodes. You might have cortisol go up compared to what's normal, or neuronal activity go down, or BDNF go down. But at the end of the day, you're going to swirl around in this basin and drop back down into this healthy basin. And so the idea of disease with this model is that what can happen is that you can undergo these perturbations that actually kick you out of this healthy spot and throw you into an unhealthy basin. And what that means is that you're going to be stuck in a place where levels of the different parts of this pathway are not healthy. And even if all perturbations are removed, you're just going to drop back down to this unhealthy basin where you have, dysregulation in a number of these different markers. And so the idea of a treatment with this idea is that you would take this current state where this person is, and you devise a way to flip whatever's broken or dysregulated and throw it back into this healthy basin. And so if we use the neuroinflammation network as an example here, what that would mean is, let's say we know that a patient has high levels of pro-inflammatory cytokines, high levels of microglia, and low neuronal activity. And you want to devise a treatment for this, what would you do? Well, the obvious thing to do is to just flip the signature of these things to get it to normal. So you'd want to inhibit microglial activity, you'd want to inhibit pro-inflammatory cytokines, and you want to boost neuronal activity. And so then you'd have normal levels of all this. And really at its simplest level, this is what we're trying to do. We're trying to devise ways to flip things that we think are dysregulated and make them look normal. So this was done for these one or two models in the case of Gulf War illness. We want to extend this out to Parkinson's disease. So there's three main things we want to extend here. So one is to enhance the algorithms to come up with smart ways to actually find these potential interventions. So we're not just reversing a signal. Maybe there's like better computational approaches we can use to try to figure out how to fix these patients. The second thing is to not just use nodes. You can't prescribe nodes to a patient. So we want to convert these nodes into drugs or treatments so that we can give treatments to patients and test those. And the third is just to extend this out to Parkinson's disease. And if you recall from before, there's many networks that are involved, and some of these networks are very large, which has its own set of computational issues associated with it. All right, so the first thing we want to do is improve some of these algorithms. So the first thing we did is we make something. So imagine this is your patient landscape here, much like in that initial picture I have here. It's just two-dimensional. And the idea here is to get these patients to the healthy state. So the easiest thing you can do is just take the signature of what's wrong here and convert it to match exactly what's healthy. So that's the easiest way to find it. But the truth is when we get to the later issue of converting these into drugs, there's not always going to be a treatment that gets you to this perfectly healthy state. So one thing, so that's not just the only option we want to be able to use. The other thing we could do theoretically is we could explore everywhere. We could say, let's look if we take a patient and move them here, is that going to have them end up in the healthy state or here? The problem with this is that if you have really big networks, this just isn't computationally feasible. So the math on it is like the number of potential places you could go to on a landscape like this is 3 to the n. So if you get over 15 nodes, the computational complexity of this is just insane and just not really possible. So we do run some form of this with what we do now, but it's we have to set a limit on the number of like node interventions. And if you have a network that has over 100 nodes, fixing 5 or 6 things is not going to be that high a percent of the network. So what are some alternatives that we've built into this? Well, what we've tried to do is we actually map out the area of this healthy basin. So we say, okay, we could reverse the signature and get here, but other ways we could do it is we can find the closest place on the edge of this basin and take you to it. And so that just gives us other opportunities to try to get to the edge of this basin and come up with treatments. And so for this patient, the best way to attack this basin would be from this side, whereas for this patient, it's over here. So it provides a little bit different, treatment options for individual patients to get back to this healthy state. All right, so we've tried to improve this way to detect potential interventions. The next thing we want to do is we want to end up with therapies, drugs or interventions, not just nodes that we know we need to modify. And so we've looked through a lot of publicly available data and also some internally generated data on information about drugs and their known interactions. And we've compiled a library of drugs and signatures. And what I mean by signatures is we look through a drug and we basically make this long list of all the known interactions that this drug is known to have. So this drug might increase BDNF, it might decrease cortisol, it might do, a number of different things. We list out every possible thing that we know about those drugs. And then when it comes time to give an intervention, so if we have a patient state and then we want to get them to this post-treatment state, we know we need to modify this node up and this node up. We go through our list of drugs and we say, okay, drug 3 causes this upper node to go up, and drug 5 causes this other node to go up, and there's no conflict between these two nodes at this state. So what we need to do is we need to combine drug 3 and 5 to switch the signature. So we use this to come up with these combinatorial treatments to try to get the interventions that we want in the nodes. So now we have our better ways to search for algorithms. We have a way to end up with drug treatments for this. And now the next step is to extend this out to Parkinson's disease. And so the initial network here that I've been showing is this Neuro-Immune network, which has 13 nodes. You can populate the information from a patient from these nodes by doing an immune panel and looking at a few additional protein markers. What we've been able to do is using publicly available information and also, again, just heavily curating our own work. We've been able to identify 89 networks that are associated with Parkinson's disease that are greater than 5,000 nodes. So we've definitely expanded out what we're looking at to describe the disease. And we're populating this information with a multi-omic readout. So transcriptomics information and proteomics information, trying to fill in this stuff as best we can to know what's changing in these patients. So just again, a breakdown of this. We start with the blood samples, we get the PBMCs, we convert those into IDANs, we get the omics readout, we fit the omics readout onto these 89 different mechanistic networks. We throw the information from our drug library in there in order to simulate how to fix what we think is broken within these networks. And then we get this sort of like step-by-step process of, trying to fix this. And this is how we come up with our personalized treatments. All right. So where we are, we have the we have the patient data that we think is representing the problem. We have these computational approaches. Now, how are we going to test these? So because we know all these networks and the genes that we believe are dysregulated and the combinations, what the combinations are actually going to fix, we can actually take all this information and we can map it to assays that the lab can measure. So we turn around and we talk, I'll talk to Matt and I'll say, okay, we have all this. We're able to figure out that, for example, that Neuro-Immune network is dysfunctional, and we think you can capture information about what's fixed by giving combination 4. By looking at this track A, track B kit, we expect the levels of track A and track B to increase based on what we've seen here. So they'll know that the network was dysfunctional, that the network is supposed to be fixed by a therapy, and they know that they can buy these commercially available kits to actually test whether or not our predictions are coming true. And so, for example, in this example I'm giving here, that Neuro-Immune network was actually dysregulated in combo 4, which actually I think it's this orange dot here. I blocked the names of the networks, but that's the network that was dysregulated. So here, before any sort of treatment in this patient, this Parkinson's patient, before they had any treatment, we had this level of dysregulation across the Neuro-Immune network. When we gave this combinatorial treatment, we see that the dysregulation is removed. We predict that the dysregulation is going to be removed. We provided this information to the lab and we asked them, hey, could you please test this and see if this is true? And so they devised this experiment where we started with Parkinson's disease IDANs, and we looked, it says day 0 here. It's really day 60 of development of these, but it's day 0 of the experiment. They gave a vehicle, so a placebo, nothing, essentially, just the carrier for what the drug is ultimately delivered in. They give it to these dopaminergic neurons, and we look one day later, and we did RNA-seq on this, and we looked at the output, and we said, were the changes that we saw of dysregulation at day 0 still present at day 1? And we did the same thing with these IDANs, except instead of giving the vehicle, we gave this combinatorial treatment. So something that we think is going to fix the Neuro-Immune network. And we asked them to do RNA-seq at day 1, and then also at day 3. And so when we did this, we have the patient, we give the vehicle, and prior to the vehicle treatment, there was 31% of the network was dysregulated. 1 day post-treatment, again at 23 to 38%, so almost no change in dysregulation was observed. And our prediction of if this was actually, if we had moved them into the healthy base and if they were ultimately going to be fixed from this, was no different or even worse. So again, there was no fix here from the vehicle. When we gave the combination, we saw 23 to 15, 23 to 0. So either the network was completely fixed at 1 day post from the combination, or it was 23%. And we predicted that we'd actually moved 2 of these 3 replicates into the healthy basin itself. And when we looked 72 hours later, again, the treatment was improved and the predicted future dysregulation should drop to 0 over time, is what we found here. Additionally, the lab went ahead and they tested this combination on an oxidative stress assay, which we also predicted to be fixed by giving this treatment. And when they looked at this combination in different doses, they saw that there was this big reduction in ROS, which, if you remember earlier, there was an increase in ROS for the PD cell line. So this other mechanism that we think should be fixed was improved. So where we are, we have patient data where we can reproduce what's wrong. We have this way to come up with combinatorial treatments and fixes. And we have some promising data right now that's showing that in these patient cells, there's improvement in the mechanisms we think we're fixing. So where do we want to go from here? So this is a heat map that shows all of the patient data that we have with all of the networks that we're investigating here. And this is organized by what's called hierarchical clustering, where samples and networks that are similar are grouped together. And so in the middle here, this is actually, you can see, so the brighter the color, the more dysregulated the network is. And in the middle here, there's a big clump of control patients, so patients that don't have Parkinson's disease, and there's mostly nothing wrong with these patients, as you would expect. But what we've noticed when looking at all this patient data is that you have sort of these two sort of different pathological cohorts. So there's one group that has a certain pattern of dysregulation down here amongst networks that are at the bottom here, whereas this other pathological cohort has an even stronger relationship of these networks here, but also this like band of network dysregulation at the top. So what we're sort of hoping to do with this information is try to subtype the disease. So maybe we can start to put together information about, not stuff to just individually treat a patient, but we can come up with therapies that will work more robustly in targeting these networks that are commonly dysregulated amongst these patients, so we can have more widespread therapies across these different cohorts. So in summary, we've created a personalized approach for therapeutic suggestions to treat Parkinson's disease. We're utilizing patient-inducible dopaminergic neurons. We're utilizing logic modeling and additional computational techniques, trying to validate this in patient samples. We're continuously, all the time, trying to iterate and improve the way we're making these predictions. And our goal is to move this to the clinic. Like if we can get really positive results in many different patient cells, we want to move this forward. We want to try this in people. So I want to say thank you. There's a lot of people at our company that have been involved in this research. So, the whole data science team, a lot of the graphics you've seen here come from Kip and Iggy, bioinformatics, who are responsible for processing the omics information, which is Anik and Edris, and the in vitro lab, who provided a lot of the results that you've seen in this presentation, which, again, is led by Madeline. And I think a lot of the results that I've seen today have come from Ritika, Erica, and Ashwarya are the people who've done there. And then again, there's just a lot of other additional. We have a modeling team as well who's been critical for the algorithmic side of this. So it's contribution of many people is the point I'm trying to get across. So thank you, and questions.